Welcome back to the deep dive. Today we're plunging into something, well, pretty fundamental to modern medicine, but maybe something you haven't thought too much about. How our medicines actually fight off tiny invaders.
Right, we're talking about antimicrobial therapy.
Exactly. And our mission really is to unpack the sheer ingenuity behind how these drugs manage to take out bacteria, fungi, viruses, parasites.
Without taking us out in the process.
Precisely. It's this incredible microscopic battlefield, and we want to give you the insight track on how it all works, especially this idea of selective toxicity.
And what's truly fascinating here is that core challenge. How do you kill something that might look biologically quite similar to your own cells? Or even trickier, something that actually gets inside your cells and uses them to survive?
Yeah.
So this deep dive, it's really about uncovering those clever biological distinctions, those little vulnerabilities scientists have figured out how to target.
Absolutely. And that challenge leads us straight into the first big idea, selective toxicity. I mean, you take an antibiotic, right? Have you ever wondered how that little pill knows to kill the bacteria causing your strep throat but leaves your own cells alone?
It's not magic, definitely not.
It's this principle. Selective toxicity. It's absolutely fundamental to making these drugs work safely.
And here's where it gets really interesting. As you said, it all hinges on basic biology. Our cells, human cells, are eukaryotic, okay? But bacteria, which are the targets for so many common drugs, they're prokaryotic. Think like a fundamentally different blueprint, A.
Whole different operating system.
Almost exactly. And that massive difference gives us targets. It's why, generally speaking, antibacterial drugs can sometimes have fewer side effects than, say, antifungals.
Because you hear too like us.
Precisely. So the targets are harder to find without risking hitting our own cells.
So selective toxicity isn't just a nice to have. It's the absolute bedrock. It means finding and hitting something the pathogen has, but you don't.
Exactly. And if we connect that to the bigger picture, you see why different drugs work for different things. For bacteria, being prokaryotic, you've got unique features, maybe the cell wall structure or specific metabolic pathways. We just don't use plenty to aim at.
Right.
But then you look at fungi or parasites, they're eukaryotic, much closer to us, biologically finding those unique targets without causing collateral damage to the host, to you, that's a major hurdle.
Okay.
And then you've got viruses, the ultimate challenge. Maybe they're obligate intracellular pathogens, meaning they
have to get inside our cells to reproduce. They hijack our machinery.
Totally. They don't have their own factories, they use yours. So treating a viral infection often means interfering at least a little with your own cell processes that the virus needs. It's incredibly complex, which explains why an
antibiotic for a bacterial infection does absolutely nothing for your cold, which is viral zero.
It's looking for bacterial targets that simply aren't there. In a virus, the scope of activity is highly specific.
That makes perfect sense. So, okay, let's focus on bacteria first. They're probably the ones we encounter most often. In terms of infections needing treatment, how did we even start figuring out how to fight them?
Well, the classic story, right?
Alexander Fleming penicillin, the moldy bread discovery. Or rather, the moldy petri dish.
Exactly. A bit of luck, a lot of observation, and it highlights something important. Many of our early and even current antimicrobials actually come from other microorganisms. Nature's own arms race.
So we find something that kills bacteria. What does it actually do? You mentioned cell walls earlier, being different, right?
That's a huge one. Think of bacteria as having different types of armor. You've got gram-positive bacteria. They have this thick, strong mesh layer called peptidine glycan, really robust.
Okay.
Then you have gram-negative bacteria. Their peptidyl glycan layer is much thinner, but they have this extra outer membrane, kind of oily with lipids and stuff like lipopolysaccharide. It's a different kind of defense.
And these defenses matter for treatment?
Absolutely. Some drugs can get through one type of wall, but not the other. It dictates which drug might work.
Okay, so when a drug does work, what's the outcome? Does it just kill them off?
Well, there are two main ways these agents work. Some are bactericidal. They actively kill the bacteria. Penicillin's a good example.
Kills them dead.
Right. Others are bacteriostatic. They don't necessarily kill the bacteria outright, but they stop them from growing and multiplying. Tetracycline is like that.
So why wouldn't you always want to just kill them? Bactericidal sounds better.
But sometimes stopping them is actually preferable. Think about sepsis, a massive body-wide infection. If you use a powerful bactericidal drug and kill vast numbers of bacteria very
Quickly they break open.
exactly, and they release toxins like endotoxins from gram-negative bacteria. This can actually trigger a huge inflammatory response, potentially making the patient even sicker in the short term. So sometimes a bacteriostatic approach, just holding them in check while the immune system catches up, is gentler.
Huh, never thought of it that way. Are there other strategies?
Oh, definitely. We use synergy. Sometimes that's when two drugs together work much better than either one alone.
Like a tag team?
Kind of. A classic example is combining an aminoglycoside, which messes with protein production inside the bacteria, with something like a penicillin, which weakens the cell wall. The penicillin basically punches holes so the aminoglycoside can get inside easier. We also talk about the spectrum of activity. Broad spectrum antibiotics hit a wide range of bacteria, both gram positive and gram negative. Useful when someone's really sick and you don't know exactly what bug it is yet. It's called empirical therapy.
Covering the bases.
Right. But once you do identify the specific bacterium, maybe from a lab culture, you ideally switch to a narrow spectrum antibiotic, one that targets just that specific type or group of bacteria.
Why narrow it down?
One, it's often more effective against the specific target. Two, and this is crucial, it reduces the risk of promoting antibiotic resistance. Using broad spectrum drugs unnecessarily wipes out lots of your good bacteria too, and gives resistant ones more space to grow.
Ah, right, the resistance problem. So identifying the bug first, evidence-based therapy is key.
Absolutely. It leads to better outcomes and helps preserve our antibiotics for the future.
Okay, let's unpack some of these mechanisms then. How do they actually work at the molecular level? You mentioned cell walls.
Yep, cell wall inhibitors are a major class. Think penicillins, cephalosporins. They mess with the enzymes, the little protein machines that bacteria use to build and maintain that strong peptide dog-like in wall.
So the wall just falls apart?
Pretty much, or it can't be built properly in the first place. The bacteria basically rupture and die, especially gram-positive ones that rely so heavily on that thick wall.
And vancomycin, I hear about that for serious infections.
Right, vancomycin is a glycopeptide, also a cell wall inhibitor, but it works slightly differently. It stops the building blocks from even being added to the wall. It's often a last resort for resistant infections like MSSA.
Are there side effects with these?
There can be. Allergies are common with penicillins, from rashes to rarely severe anaphylaxis. Some, like vancomycin, need careful monitoring because they can potentially affect the kidneys or hearing. And sometimes you see resistance where bacteria produce enzymes like beta-lactamase that chew up the penicillin.
So we have drugs to fight the resistance.
We do. We have belactamase inhibitors like clavulanic acid, which you find in augmentin. It doesn't kill bacteria itself, but it stops the bacteria's defensive enzyme, letting the A-cylinder partner do its job.
Okay, so that's the wall. What else can we target?
Another huge target is protein synthesis. Bacteria need to make proteins constantly to function.
Like tiny factories.
Exactly. So drugs like aminoglycosides like gentamicin or tetracyclines or macrolides like azithromycin, they target the bacterial ribosome. That's the protein-making machine.
But we have ribosomes too, right? To make our proteins.
We do. But here's the selective toxicity genius again. Bacterial ribosomes, prokaryotic ones, are structurally different enough from our eukaryotic ribosomes that these drugs can bind specifically to the bacterial ones and shut them down, leaving ours mostly untouched.
That is clever. Any specific concerns with these?
Aminoglycosides are powerful, but they're tricky. They're cleared entirely by the kidneys. So you have to check kidney function and adjust the dose carefully. They can be nephrotoxic, damaging to kidneys, and also ototoxic, meaning they can affect hearing or balance.
Wow. Okay, so careful monitoring needed there. What other bacterial weak spots are there?
We can go after the nucleic acid synthesis, DNA and RNA. One really elegant example involves folate synthesis.
Folate, like in vitamins?
Yeah, folic acid. We humans get it from our diet, we have to eat it, but many bacteria, they have to make their own from scratch.
Ah, another difference to exploit.
Exactly. So drugs like sulfonamides and trimethoprim act like roadblocks in that bacterial folate production line. They competitively inhibit key enzymes. The bacteria need no folate, no building blocks for DNA and RNA, no growth.
And it doesn't hurt us because we just eat our folate. Brilliant.
It is. Other drugs in this category might directly interfere with enzymes like DNA gyrase, which bacteria need to manage their DNA during replication, and reading has side effects for these, mostly things like stomach upset or allergies. But sulfonamides can sometimes cause crystals to form in the urine, which could potentially harm the kidneys if the patient isn't well hydrated.
Okay, and just quickly, what about really tough bacteria like the ones causing tuberculosis?
Right. Antimicrobacterial drugs are a special case because mycobacteria have unique waxy cell walls. Drugs like isoniazid, we're actually still not 100% sure how it works, but it's effective. And rifampicin, which specifically inhibits the bacterial version of RNA polymerase, stopping them from reading their DNA instructions.
Complex stuff. So, all right, we've covered bacteria pretty well, but what about those trickier eukaryotic targets? Fungi and parasites. You said they're more like our own cells. How on earth do we target them? Selectively.
Yeah, that's where the challenge really ramps up. But scientists have found ways for antifungal medications. One key difference lies in the cell membrane.
How so?
Fungal cells use a lipid called ergosterol as a major component of their membranes. Our cells use cholesterol. It's a subtle but crucial difference.
So the drugs target ergosterol?
Many do. Some bind directly to ergosterol, punching holes in the fungal membrane. Others inhibit the enzymes, often specific cytochrome enzymes that the fungus needs to make ergosterol in the first place. No ergosterol, the membrane integrity fails, the fungus dies.
Still exploiting those small differences?
Exactly. Other antifungals might interfere with DRNS synthesis, again, looking for subtle variations in the fungal enzymes compared to ours. And of course, for topical antifungals you put on skin, selectivity is maybe less critical because it's not getting deep into your system.
Makes sense. And parasites? Same kind of challenge?
Very similar challenge, yes. With antiparasitic drugs, the strategies often involve finding enzymes that are absolutely unique to the parasite. If it doesn't exist in humans, great target.
But what if there aren't many unique ones?
Then it gets harder. Sometimes you have to target an enzyme that both the parasite and host have. But maybe it's much more critical for the parasite's survival than for ours. Or perhaps the enzyme works slightly differently in the parasite, allowing a drug to inhibit the parasite version more strongly. It's a fine balancing act to hit the parasite hard without causing too much toxicity in the host.
And this brings us back to patient differences, right? You mentioned an example with antimalarials.
Yes, a really important one. G6PD deficiency, glucose 6-phosphate dehydrogenase deficiency. It's a genetic condition affecting red blood cells. Certain antimalarial drugs like chloroquine or primaquine can trigger severe destruction of red blood cells. Homolysis in people with this deficiency. It's a stark reminder that it's not just about the drug and the bug. The patient's individual biology is absolutely critical. You have to consider these things.
That really hits home. How complex this is. Okay, final category. And maybe the toughest viruses, the hijackers. They live inside our cells. How do we even begin to fight them without wrecking our own cellular machinery?
It is arguably the most intricate puzzle. Because viruses are using our equipment, antiviral drugs had to be incredibly specific. They aim to interrupt the virus's life cycle at first, very particular points.
So stopping them from getting in, maybe?
Exactly. Fusion inhibitors do that. They block the virus from latching onto and entering the host cell. Stop it at the doorway.
Okay, what if they're already inside?
Then you target their replication steps for viruses like HIV that need to convert their RNA genome into DNA. You have reverse transcriptase inhibitors. Some mimic the building blocks, nucleosides, gumming up the process. Others directly disable the enzyme itself.
So sabotaging their copying machines.
Precisely. Then there are protease inhibitors. Viruses often make long chains of proteins that need to be cut up by a protease enzyme to become active. These drugs block that enzyme so the virus can't assemble new functional virus particles.
Stopping the assembly line.
You got it. Others, like viral DNA polymerase inhibitors, directly block the enzyme that copies the viral DNA for certain types of viruses, or viral code disassembly inhibitors, stopping the virus from properly unpacking itself once inside the cell.
It's like finding all these little choke points in their plan.
Exactly. And sometimes we don't attack the virus directly, but boost our own defenses. That's where biologics and immunomodulators like interferons come in. They help ramp up the body's natural immune response against the virus.
But ultimately, because they use our machinery.
Effective antiviral therapy often does involve inhibiting some host cell functions, at least temporarily or to a certain degree, to disrupt that viral replication cycle. It's always that delicate balance: efficacy against the virus versus safety for the host.
So, pulling this all together, what this deep dive really shows is just the incredible cleverness and the sheer variety of strategies involved in fighting microbes. We've gone from exploiting really obvious differences like bacterial cell walls to finding these tiny metabolic quirks and fungi and parasites, and then figuring out how to disrupt viruses, literally hijacking our own cells.
It's a constant scientific endeavor, this balancing act between killing the invader and protecting the patient.
Right, and if we look at the bigger
Picture here, well, it underscores how dynamic this field is. New therapies are always being developed. Research is constantly ongoing, always aiming for better outcomes, fewer side effects. It also really highlights the complexity, the need for that evidence-based approach, using the right drug for the right bug, and remembering those crucial patient factors like G6PD.
definitely.
So maybe a final thought for you to consider: given how incredibly fast microbes can adapt, mutate, develop resistance to these amazing drugs we've developed, what responsibility do we all have globally to use these powerful tools carefully, wisely? How do we make sure they remain effective for generations to come?
Well, that's where we'll wrap up today's deep dive. If you found this interesting, give the video a like. It really helps the channel grow. And don't forget to subscribe so you don't miss the next episodes. Thanks for watching, and see you in the next video.